So without further ado, I would like to introduce our next speaker, our keynote speaker, Dr. Tom Boyce. Dr. Boyce earned an MD from Baylor College of Medicine. He's a pediatrician, completed his residency at the University of California, San Francisco, and he spent 20 years at Berkeley as a professor of epidemiology and child development, an associate dean for academic affairs and research at the Berkeley School of Public Health. He's a leading expert on the interplay between neurobiology and psychosocial processes. His work has demonstrated how psychological stress and neurobiological reactivity to adverse social contexts operate conjointly to produce disorders of both physical and mental health in child populations. Tom is a real proponent of the importance of oral health as sort of a view to what else is going on with general health, and so we welcome Tom to talk about his work. Thank you, Tom.
Thank you, Karen, for inviting me to be a part of this terrific symposium. I am a great proponent of anything that bridges the pond out here between UCSF and Berkeley, having spent a substantial amount of time on both campuses and realizing how much commonality and how much could be gained by bridging collaborations between the two campuses. I'm going to place our consideration of oral health disparities in an even broader context. In fact, a context maybe so broad that you'll wonder if the wrong speaker wandered into the auditorium and somehow got up here on the podium. But if you'll bear with me, I think I can make the case that there actually is something that I've been doing that has some relationship to the importance of oral health, both here and worldwide. And I want to place it within this broader context by asking how inequalities in human societal structures generate stress and adversity, even in early life, exploring how that adversity may play a role in differential susceptibility to both childhood caries and also to other morbidities, and how new research on gene-environment interplay may inform that differential susceptibility and illuminate the ways in which childhood caries may be linked to disorders of both biomedical and mental health within children and on into adult life. So a fairly expansive agenda, and I hope that I'll be able to succeed in persuading you that it may be that dental caries, and in fact the mouth itself, may be a kind of window onto the developmental origins of disease within adult populations and within childhood populations. So thus this title of An Oral Biology of Misfortune. I think you'll begin to see what I mean by a biology of misfortune. So my work really begins and began many years ago with this most well-replicated finding in all of Child Health Services research, which is that within any childhood population that you want to choose, anywhere around the world, there is this highly uneven distribution of morbidity within that childhood populations. So if you look at these little darker individuals here, they constitute about 15 to 20% of any given childhood population, and they represent well over half of the biomedical and psychiatric and I would argue probably dental morbidities within the entire population, and are responsible for well over half of the healthcare utilization within that population. So there is this tremendously uneven, non-stochastic, non-random distribution of morbidity within childhood populations. And that turns out to be a really fundamental observation, because it means that there is not just differences in rates of morbidity between populations, but even within populations, even within the population of the San Francisco Bay Area or San Francisco County or whichever population you want to choose. There is this highly divergent, highly uneven distribution of disease. So really it means that we need to address what I think of as these sort of profound two twin human mysteries. And those are the mysteries of why some get sick and others do not, and what the origins are of our differences, those kinds of differences that we saw in that last slide. So we have been learning, working, practicing over the last 3 or 4 decades within a period of tumultuous reorganization and revision of our understanding of the origins of disease etiology. When I was a pediatric resident at UCSF in the 1970s, we were in a period of extreme environmental determinism. When I rotated through the family therapy unit at UCSF, I was actively taught that autism was probably related to something having to do with cold, withdrawn parenting, with a special focus on mothers. And it was a period of time when the concept of schizophrenogenic parents was very much at play. And it was really an environmental determinism that was responsible for a really unfair, mistaken, misleading kind of thinking about the etiology of various pediatric disorders. We then, with the advent of the Human Genome Project, entered a period of what I would argue was genetic determinism, where we were dead certain that there was going to be a gene for schizophrenia, a gene for autism, perhaps a gene for dental caries. And we were in a period of time. Anybody want to get connected to the internet? We were in a period of time where genes were really thought to be the principal driving etiology of most morbidities. We then entered a period of time where we began to partition variants in behavior genetics into that portion that's related to environment and that portion that's related to genes. But again, it was kind of a separation of genes and environments. Then we began to discover about 10 years ago with the publications of Caspi and Moffitt and others that there actually were gene-by-environment interactions that we were discovering within the epidemiology of human disease. We began to see linear regression equations like this, where cross-product terms representing the effect of genes by environments began to appear. And it really was only recently, only in the last several years, that we began to understand that really much closer to the truth of all of this may be that there is a kind of gene-environment interplay in which epigenetic processes play a key role. And that's the story that I want to talk about today. So it is a story in 2 parts from my own work. The first part has to do with neurogenomic differences in susceptibility to social adversity. I'm going to argue that stress and adversity is a prominent factor within childhood populations, within the lives of some children, and that it actually plays an etiologic role in certain kinds of disorders, among them dental caries. The second part of the story has to do with oral, biomedical, and mental health effects of social subordination. This stratification of human societies that turns out to happen both at the level of human societies and down at the level of small human social groups. So this is where I'm headed, these 2 stories. So let me begin by making the observation that children who grow up in blighted, really disadvantaged circumstances have far greater rates of both acute and chronic disease as you follow them over time. That is the genesis of this now iconic relationship between socioeconomic status, here going from low to high, and rates of all kinds of physical chronic diseases within children, including any limiting condition, ear disease, physical inactivity, asthma, injury. As you can see, there is this inverse linear monotonic association with kids at the highest levels of social class having far lower levels of these chronic disorders than children who are at the low end of the SES spectrum. It's interesting to note that this is not what my Canadian colleagues would call a hockey stick association. It's not just that it pops up here in the very low, most impoverished end of the spectrum. It is, in fact, that at every level of social class, there is slightly higher levels of chronic disease, even at the high reaches where these are children of professional class families, and these are children of the very, very rich. Now, we think of all of these kinds of etiologic factors when we talk about socioeconomic disparities in child health and development, including environmental exposures to physical toxins, dietary differences, healthcare, housing, exposures to violence both within the home and within the neighborhood, parenting differences, and differences in exposure to adversity and stress. We also know that childhood socioeconomic status is not simply related to differences in morbidity within childhood concurrently, but actually casts a shadow on into adult life as well. So here we show this is cumulative morbidity from coronary heart disease among children from adults with low childhood SES experiences versus high childhood SES experiences in the age range from 35 to 75. These are actual trajectories of cumulative morbidity, and it shows that the poor children have higher cumulative rates of chronic disease morbidity all the way through adulthood. And this is true not just for coronary heart disease, but for a variety of 20 minutes, really?
No, we got started.
Okay, I will speed up here.
Tom, we can cut into our posters.
Okay. So what are some of the explanations? Here I go to join AT&T again. What are some of the explanations for these kinds of differences? Well, first of all, we know that there is a socioeconomic partitioning of stressors and adversities. And we find that if we compare children growing up in poverty, children growing up in middle-income families, this is from the work of Gary Evans at Cornell, that among children in poverty circumstances, there's much greater density of housing. There are other density or housing problems. There's a far greater exposure to noise in the ambient environment. There's more family turmoil and chaos, and there's far greater exposure to violence within the home and outside of the home. We also know that these important adverse childhood events that have been studied by Vince Felitti and colleagues, they show that among the leading causes of adult mortality, coronary disease, diabetes, sexually transmitted disease, drugs, and suicide, that if you enumerate the number of adverse childhood events, that in each of these circumstances there is this graded association. So this, again, projection of these experiences of stress and adversity on into risk within adult life. We also know, however, that there are more mundane quotidian kinds of differences in the lives of disadvantaged and advantaged children from differing communities. This is the celebrated work of Hart and Risley, who in 1995 published their book on differences in the everyday lives of American children, showing that when you compare welfare, working class, and professional class families, that the average number of parental utterances to the child per hour was systematically different in this, again, sort of linear monotonic association. They were the ones that showed that by age 3, there was a 30 million word gap in the vocabulary experience of children growing up in welfare families versus growing up in professional class families. The more recent work of Anne Fernald at Stanford looked at processing efficiency. She put stories in front of children, asked them, where's the doggy or where's the cart or something like that, and looked at the actual processing time of the children looking to the target word. And she found that there was equivalence between children from high SES communities at age 18 months and children from low SES communities at age 24 months. So even by 2 years of age, a 6-month gap in the ability of children from low SES communities to process language and respond accordingly. So it turns out, in a nutshell, that there are just these pervasive differences in the experiences of everyday life among children growing up in poor communities versus those growing up in middle class and upper class communities. But it also is true that there is a tremendous amount of variability in the effects of these social factors on outcomes that we care about. So, for example, these are old data of mine, looking at behavior problem severity scores as a function of family stress. And you can see that there is a linear monotonic association, but you can also see that when you actually look at the plotted individual data points, that there's a tremendous amount of variability in the character of this association. These are Canadian data on children from different social class groups on literacy and reading scores. And again, this very powerful association that we are accustomed to seeing, but when you actually look at the data, the individual data for individual children, you see that there are children from very high socioeconomic strata that are having very low reading scores and literacy scores, and similarly children from low SES families who have exceptional literacy scores. So stressful, disadvantaged environments have these highly variable effects on developmental and health outcomes. Years ago, we began studying this variability by starting to question whether this variability within these associations was really the noise, or might it be the music, and might that be precisely what we ought to be focusing on. So years ago, now measured probably in decades, my colleague Abby Alcon, who's sitting right over there, from the UCSF School of Nursing, and I began bringing children into very standardized, previously unknown circumstances, where they were placed in front of an unknown examiner and given a series of mildly stressful tasks in a variety of different domains. We did things like ask them to repeat digits, the span of digits back, had them look at emotion-evoking video clips, had them interviewed by this previously unknown examiner. And we began to try to index in these very scripted, very prescribed, standardized circumstances the responses of these children to the stressors that they were experiencing. And we used, in indexing stress response, the two principal stress response systems within the human brain. One is the CRH system, the corticotrophin releasing hormone, which begins here in the hypothalamus, signals the anterior pituitary to put out ACTH, which then produces cortisol, so the so-called HPA, hypothalamic pituitary adrenocortical axis. The other is the autonomic nervous system, which really begins here in this little nucleus in the brainstem, which has dopaminergic projections up into the hypothalamus and ignites the whole panoply of fight or flight responses that we're all familiar with. The sweaty palms, the tremulousness, the dilated pupils, and so on. And we used these measures of both autonomic and adrenocortical reactivity in these highly standardized laboratory circumstances. We used impedance cardiography to measure autonomic response and salivary cortisol levels to measure adrenocortical response. And we found in a nutshell that there were these broad, reliable individual variabilities in the magnitude and patterns of response that we saw within children. What we then were able to do was to take these laboratory data on where the children lay in this continuum of stress reactivity and use those within epidemiologic study designs where children were being subjected to naturally occurring stressors in the lives of their families and communities. So we have now amassed quite a literature of replicating studies, and I'm going to show you just one because it's nicely exemplary of the kind of things that we've been finding for the last 15 years or so. So this is from one of my postdocs, Jelena Obradovich, who's now an assistant professor at Stanford. She studied pre-syndromal psychopathology in the lives of children who were living in families with either low or high levels of marital conflict. And she divided the sample of children into those who in the lab had shown low autonomic nervous system reactivity or those with high autonomic nervous system reactivity. The high children being those that were at the top 15 or 20% on that continuum of ANS reactivity. And here's what we find. We find that, first of all, that the great vast bulk of the children who are low in autonomic reactivity to these mild standardized stressors basically are indifferent to the level of marital stress that they're experiencing within their homes and households. But by contrast to that, when we looked at the high reactivity children, we found, as we had hypothesized, that at the high stress end of the scale, that these were children who had the highest levels of psychopathology of any of the children within the samples that we studied. What we had not anticipated, and what we have found repeatedly now, over and over again, is that the high reactivity individuals in circumstances of low marital conflict have the lowest rates of psychopathology when they are living in families where marital conflict is relatively rare. So they had either the worst or the best of the outcomes depending upon the kind of social context in which they found themselves. Our account for that, our explanation, is that there is a kind of differential neurobiologic susceptibility to social context. It appears that there are children who are highly sensitive to the effects of both negative and positive social contexts, both within the family, within the schools that they attend, and within the communities in which they are embedded. For shorthand, we have used this wonderful Swedish word, an idiomatic expression, maskrosbarn, which means dandelion child, to refer to the kids who, like dandelions, can seemingly grow wherever you want to plant them. Dandelions grow in fertile fields, they grow in the cracks between sidewalks, they grow at altitude and in valleys. They really grow wherever you want to put them. And it appears that most children have this kind of profile of being dandelion-like. In contrast to that, we coined this Swedish neologism, orkidebarn, which means orchid child, for these kids who, like the orchid, are exquisitely sensitive to the character of the social environment in which they are being reared and have either the worst outcomes or the best outcomes depending upon those circumstances. We have also found the same kind of interaction at the level of genes. This is some work by one of my ex-postdocs. AT&T, you can't, it's sort of like Coca-Cola, I guess. You can't get rid of it. But here again, these are children in the BDNF gene, brain-derived neurotrophic factor, who are either MET carriers or VAL-VAL homozygous. And again, we find that there are these orchid children who have either the highest output of cortisol within a given day or the lowest, and the other children, the dandelion children, who are relatively indifferent to the effects of family income, which is shown here on the horizontal axis. It also appears to work with regard not just to morbidity, but to developmental events as well. This is going to be a plot of Tanner staging, a measure of puberty. We all begin as Tanner 1s, and we all end up as Tanner 5s. But it turns out that the trajectory, the developmental trajectory by which you get from 1 to 5 is a pretty important thing, because it provides risk or protection from things like early sexual debut, adolescent pregnancy, acquisition of sexually transmitted disease, and so on. And if you can delay the onset of puberty, it actually protects you from these kinds of morbidities. So here again are these children divided into 4 groups. The reds are going to be those with low autonomic reactivity, sympathetic nervous system reactivity. The blues are going to be those who are high in reactivity. The closed data points are those from low parental warmth families, and the open data points are those whose families are characterized within observations by high parent-child warmth. The horizontal axis here is centered on 12 and a half years. It goes back 3 years and forward 3 years. So you're going to see the trajectories of pubertal development among kids of these different kinds. First, we show that kids who are low in sympathetic nervous system reactivity have approximately the same very linear growth in pubertal development from stage 1 to almost stage 5 over this 6-year period of time. The kids who were low in parental warmth in parent-child observations, but who were high in sympathetic reactivity, had this accelerated rate of pubertal development. Again, everybody ends up in the same place, but it's this difference in slope that turns out to be important. And the kids who were equally high in sympathetic reactivity, but who had highly warm, highly demonstrative parents, didn't even go into puberty until age 12 and a half, and then caught up. So here again, we have the dandelion and the orchid. The orchid children who have either the worst trajectory or the best trajectory, depending upon the character of the parenting environment in which they are being reared. So we have this phenomenon of differential neurobiologic susceptibility to social context happening at the level of behavior, at the level of brain development, at the level of synapse and circuitry development within the brain, and even at the level of genome and epigenome. So a question might be, what about at the level of society, farther up this scale? Is there any kind of indication of this same phenomenon at the societal level? So here I come to the second of these 2 stories, which is the oral, biomedical, and mental health effects of social subordination. So here is where my own work converges with that of the other speakers and many of the audience, with the issues of oral health and disparities in oral health among children of different groups. Again, to repeat what you've heard from many of the other speakers thus far, dental caries are the single most common chronic disease of childhood. It has very strong SES and racial disparities. There are known etiologic factors involving lead exposure, tobacco smoke, diet, dental hygiene, and access to fluoridated water. We know that the oral bacteria, Strep mutans and Lactobacillus acidophilus, are implicated as essential but not sufficient etiologic factors. And the word on the street, at least among pediatricians, I don't know about pediatric dentists, is that the leading account of why there are these socioeconomic and racial disparities in childhood dental caries is that there is a neglect of children's dental hygiene by low SES parents. So we set out to try to probe that, what we believe now is a misunderstanding, at a deeper level. So in our studies of young children, as you've heard already, one of the things that we have wanted to do is to tap into the adrenocortical system as a measure of response to the everyday stressors of life. And the way that has typically been done, here's what you probably know is the typical diurnal pattern of cortisol secretion. Cortisol peaks just as we're waking up in the morning. It then follows a descending path, and just about the time we're going to bed at night, it reaches a nadir. And what we would really like to know is what's the overall area under that curve. What we're relegated to looking at usually is these 3 different time points where we measure cross-sectionally the level of salivary cortisol within the child's saliva, despite the fact that what we really would like to know is this chronic exposure to cortisol, the area under the curve, if you will, not just within a day, but over several days of time. So we were pondering a few years back how we could get a bioassay, a biomarker of chronic exposure to higher levels of cortisol. And we thought about Cushing's disease, where we know that there is pathologically high levels of cortisol that are sustained by usually adults with this disease. And it leads to osteoporotic processes that can lead to collapsed vertebrae, demineralization of calcified tissue, and so on. And we thought about that for 5 minutes because obviously we weren't going to X-ray children's back or feet or whatever. But then we started realizing that the very subjects that we were most interested in, which were 5-year-old children in kindergarten, that they were individuals who were going to be losing a calcified tissue out of their mouth within the next year as they entered first grade. So we thought, wow, maybe this is a chance to assay cortisol exposure over time. So we began to consider the question, could deciduous teeth be used as a stress biomarker in young children? We were interested in whether cortisol, as recorded in the animal literature, might have effects on either the thickness or the density of these various micro compartments of dental anatomy. And we began collaborating with Pam Denbeston and her colleague John Featherstone at the School of Dentistry at UCSF, doing these wonderful little computed tomography images of these deciduous teeth that children gave to us. I should tell you, by the way, that this arm of this project was, of course, called the Tooth Fairy Project. And we paid kids $10 for these teeth, which at the time was considerably above the market value for teeth falling out in first graders. And, this went along very well. Kids were eagerly bringing their teeth in this little solution that we could then ship over to the dental school at UCSF. And one month, we got back from the dental school, we got the tooth back in this little bottle, and there was a note on it that said, "This is a dog's tooth." So somewhere in Berkeley, there is a dog missing a tooth and a kid who is illegitimately $10 richer. So we began then to use these measures of the density and character of these dental micro layers as a measure of chronic exposure to cortisol. So what did we find? These are taken from work that was published in 2010 in Social Science and Medicine. First of all, when we look at the amount of oral karyogenic bacteria in the mouth along the gingival margin, low or high, as a predictor of decayed, missing, or filled dental surfaces, and divide the children into those with low diurnal cortisol secretion versus those with high diurnal cortisol secretion, we find that karyogenic bacteria and salivary cortisol were independently and interactively associated with dental caries. You can see the difference in slope here of these two associations. And the highest rates were found in children with high karyogenic bacteria and high salivary cortisol levels. Secondly, we found here is enamel thickness measured with those little computed tomograms as a function of low versus high SES, in this case household education level, and the kids divided into those with low cortisol reactivity versus high cortisol reactivity, and we find that cortisol reactivity and SES, anybody know how to turn this off? we find that cortisol reactivity and SES interactively were associated with enamel thickness, and the lowest thickness was found in children with high cortisol reactivity and low household SES. I guess I have to...
you can keep it up, I think.
Let me just... If I just turn off Wi-Fi, will that do it?
There we go. You got it.
Okay. Thank you. So putting this all together, we argued that the linkage between socioeconomic status, which we know is a very powerful predictor of the level and intensity of dental caries within populations of children, we found really that there were two different pathways. One was that low SES seemed to beget higher and earlier levels of cariogenic bacteria in the gingival margins of the children that we studied, which we know is a contributor and was shown in the study to be a contributor to dental caries. And secondly, that the low SES children, those families were experiencing greater financial stress. They had, the children within the family had greater HPA activation, and this seemed to lead to differences in dental microanatomy, basically the anatomic resistance to these cariogenic bacteria. And most importantly, we found that the most powerful association was the interaction between bacteria and cortisol levels in predicting dental caries. So we have argued that oral health disparities are related to these two interacting pathways. One is earlier and more intensive exposure to an agent of disease among low SES children, that is the cariogenic bacteria. And secondly, more family adversity resulting in greater HPA activation, cortisol secretion, and microanatomic vulnerability. Okay. Pam, this is a branch point. I keep getting signs that
It's okay.
Are we okay?
5 more minutes. Will that work?
Okay.
7 more minutes.
Okay. Okay. Very briefly, it turns out that the social disparities in oral health or in other forms of morbidity are not just related to one's position within the large society, much of which I've been showing you, that SES is a constant perennial predictor of these various forms of outcomes. It turns out that its predictive, social position is predictive even within little human social groups. Now, why would we even study that? Well, we know that African fish maintain these severe hierarchical organizations in which dominant males only have access to the females. We know that rats in a subordination paradigm show these pro-inflammatory shifts in cytokine signaling. We know in monkeys that they form these very stable, linearly transitive social hierarchies where the subordinate ones have upregulated adrenocortical function, poor immune competence, and decreased resistance to disease. And we know, much to the chagrin of the medical students that I teach, that young children form social orders within weeks of entering new social groups. The medical students can't stand this because they think of these kids, these 5-year-olds that we've studied, as, as close to a pristine human society as you can possibly get. And lo and behold, it turns out that kindergarten teachers will tell you not only that there's a pecking order by the end of 2 weeks, but he or she can tell you exactly where every child lies. So the question then becomes, are these subordinate positions in early peer hierarchies associated with greater stress, exaggerated reactivity, and excessive stress-related morbidity? So we have conducted a study here in the Berkeley schools that we're still analyzing using naturalistic measures of dominance and subordination behaviors. We define social dominance as a pattern of repeated encounters in which the outcome consistently favors the same dyad member. We have done dominance observations involving critical event and scan sampling over 3 to 5 weeks of time within these classrooms, and we index dyadic interactions like physical attack, imitation, directing behavior, threat, relational aggression, and so on. And when we do that, we can then enter those observations into a mathematical algorithm, which results in a very clear hierarchy. You can see that there's equal representation of the genders at the high and low ends of this hierarchy, but each child occupies a place within this hierarchy in this novel social group. So I've already shown you that there is this association within large human societies. The question is whether within kindergarten societies, this was 338 children in 29 Berkeley public school classrooms, is there the same association? It turns out that there is, and what we find is that the subordinate children sustain higher rates of depression and inattention. They have poorer peer relationships, and they have poorer academic competence. And it seems to affect, in this case, boys more than girls. And this was published in a paper in PNAS a couple of years ago. I'm going to skip through this because it is not essential. So we have evidence of these kinds of differing susceptibilities now at every level of this hierarchy of complexity and scale. I will just tell you very briefly that the way that we think all of this is happening is the same way that butterflies develop these different phenotypes. These are actually the same species of butterfly, buckeyes, but they look very different, and it is a polyphenism that is driven by the temperature in the environment and the length of daylight as they emerge from their pupal stage of development. So it represents a conditional adaptation involving differential epigenetic regulation of genes determining wing coloration and pattern. For those of you who don't know what this really exciting emerging field of epigenetics is about, it turns out that there is epigenetic control of genes that guide brain development and function. Genes come, the DNA, if unraveled from the chromosome, comes packed as chromatin, and it can either be tightly packed or loosely packed. And it turns out that it's very difficult to read and express the DNA code when it's tightly packed, and it's much more easy to read and express when the DNA is more loosely packed. And the way that this happens is that there are these chemical tags that can be added either to the DNA or to the histone proteins around which the DNA is wound. And those chemical tags result from these experiences of stress and adversity and other kinds of exposures that actually determine the pattern of these marks within the epigenome. We've had a chance with the Wisconsin Study of Families and Work to longitudinally look at the influence of parental stressors reported during the children's infancy and preschool periods, go forward 15 years, and we're doing epigenetic profiling of these buccal epithelial cells in these kids, and we find that there are these epigenetic vestiges of early parental stressors. Mother stressors, shown in red, are found more related to hypermethylation of DNA sites when reported in infancy, and father's associations with demethylation, shown in blue here, primarily for girls. So a pattern of hypermethylation and demethylation that is consistent with known developmental time courses for parental influence. So it turns out that this mystery of how gene by environment interactions occur, it turns out that there is new evidence that epigenetic processes may mediate these G by E interactions. So I would like to finally argue that there is an increasingly untenable distinction between the biological and the social sciences. What we are actually seeing is this revolution in which epigenetic processes are demanding the synthesis between understandings of social environment and understandings of allelic and epigenetic variation. And so there is this kind of reciprocal interdependence of social and biological discovery, where findings within the social environmental realm are illuminating mysteries within the DNA, and differences in DNA and epigenomes are illuminating the differences that we see within social environments that we study. So, how do you want to play this nature, nurture, or a bit of both? It is definitively a bit of both, and it is interactive, it is interplay, and it is probably going to be the ultimate way that we understand the origins of dental caries, of mental disorder, of biomedical disorders extending on into adult life. So I'll stop with the question, might gene-environment interplay become a convergent principle within oral biomedical and mental health research, and might these be the linkages through mediators such as inflammatory changes and levels of microbiome within the human alimentary tract might be related to differences in brain development, differences in long-term cardiovascular disease, differences in long-term development of mental disorders. So I want to argue to you that oral health may be a gateway, really a window into long-term biomedical and mental health. Last slide, Pam and I have begun talking about what I have been, I guess facetiously, calling a Krishna stress biomarker project. There's this Hindu mythology that says that when Krishna's mother looked into her son's mouth, she saw, quote, the whole universe of moving and unmoving creation. When you think about it, the number of biomarkers that are accessible through the human mouth are truly extraordinary. You can measure in saliva cortisol, oxytocin, alpha amylase, C-reactive protein. You can culture the gingival margin for the microbiome. You can harvest buccal epithelial DNA for allelic and epigenetic variation, telomere lengths, and you can look for inflammatory changes and caries. So this might constitute a kind of portable, quick, and minimally invasive battery of oral stress biomarkers for use within epidemiologic studies, trying to illuminate and discern differences in oral biomedical and mental health. So I will stop there. Thank you.